This presentation is on stem cells, starting with the basic function and uh, potency concept in stem cells. Stem cells have two key characteristics: self-renewal, which is the ability to make more of themselves or replicate rather efficiently, and potency, which is their potential for differentiation into different types of cells. Potency is kind of like how a kid can end up in multiple different career fields, despite starting out at a relatively similar setting or level of potential. There are different levels of this potency. Totipotent stem cells can become almost any type of cell and are found in zygotic stem cells. Zygotic stem cells are an example. And this is kind of like an elementary and middle school student, where they're just developing their initial interests and don't really have any limitations on the career fields that they can go into. So an elementary school student can be developing interest that leads them to become a orthopedic surgeon or a lawyer or a railroad technician. Now, pluripotent stem cells can turn into almost any cell type. They won't turn into placental or germ cells. These are found in embryonic stem cells. Going back to the career analogy, this would be like kids in high school. High school students start to narrow down on their specialty. A kid graduating from high school can go into various different fields, but if they don't take any business classes, it's less likely that they're going to be a competitive candidate for business school, for instance. Now, multipotent stem cells can turn into multiple different cell types. These are found in adult-derived stem cells and adult stem cells more generally. Going back to the analogy, this would be like a university student or graduate school student. So, just like if you're currently studying biology, there are different career fields to go into with that degree, or studying accounting, again, various different careers. And even if you're into med school, there are various different medical careers that you can go into from there. Now, unipotent stem cells only turn into one cell type, and these are going to be your progenitor cells for specific cell types. And going back to the analogy, this is kind of like a residency or a fellowship. If you're 5 years into a neurosurgery residency, your options are pretty much become a neurosurgeon or go back to the drawing board and live in crippling poverty for an extended period of time. Now, one distinction in terms of potency that it wasn't brought up in the course material, but I found in some supporting literature, is the distinction between germ layer lineage cells. And germ layer lineage cells can derive into the germ layer specific progenitor cells. So germ layer ectostem cells, for instance, can differentiate into the ectodermal progenitor cells, meso into mesodermal progenitor cells, and endo into endodermal progenitor cells, for instance. And this is related to the different germ layers in development. All right, one source of stem cells is embryonic stem cells. These are from the inner mast of the blastocyst, as seen at that picture on the right. The blastocyst is an early embryonic stage. These stem cells are pluripotent. And in the lab, they're kept in the state of pluripotency by using transcription factors. The transcription factors used activate the self-renewal genes and suppress genes that trigger differentiation into other cell types. Then, whenever we need a certain type of cell, we can use these pluripotent stem cells to differentiate into the cell type that we need. Now, harvesting these poses ethical concerns because you effectively have to make the blastocyst inviable for this to work, which is a line that some people don't think should be crossed in science. It's an extremely controversial topic, and I won't touch on it other than that basic description that I just gave. All right, next up, adult stem cells. Now, in the adult body, the function of these is the repair and maintenance of tissue. They are generally multipotent stem cells. They're not abundant. They're found in limited numbers in specialized tissue. And in order to use these, they have to be harvested in vitro, and when using them, they have a limited potency because they're only multipotent and not pluripotent. Some examples of adult stem cells are the hematopoietic stem cells, which are found in bone marrow and make blood and immune cells. Then, in cases where there are bone marrow-based diseases like leukemia, transplant or repopulation assay may be necessary in order to replace these cells if problems arise with them. Another type is mesenchymal stem cells, which are found in various tissues and have been implicated in immunomodulation. All right, some additional materials that I found on this. I found one paper by Dr. Young, who on the previous slide there was an image by him, and in that study they found detection of cells with markers consistent with totipotent and pluripotent stem cells in rat reproductive organs. It's a pretty interesting article, and this is an extension of Dr. Young's ongoing work with high-potency adult-derived stem cells. All right, another source is induced pluripotent stem cells. So going back to that analogy I made on the potency slide, this would be like convincing a university student to drop off and pursue underwater welding. Like, yeah, underwater welding's great. It tons of benefits, good pay, and you only have to go to a trade school in order to get certified in it, with some additional training. So basically, what this process is, we take a somatic or non-reproductive cell, and then we reprogram it into a pluripotent stem cell by adding the Yamanaka transcription factors, which I've included in the slide there. And there aren't any ethical concerns, and it can be used for more personalized treatment than other stem cell types. All right, so applications of stem cells and ongoing related challenges. Cell replacement therapy is one application, which is where we replace old damaged cells with new ones. An example is therapies that are currently in development for treatment of spinal cord injury. There's tissue engineering, which can be used to make grafts of various tissues and replace cartilage. And then there's, in application, these things can be used in conjunction with gene editing technology like CRISPR in order to make treatment that's more specialized for the patient and thus more compatible with the patient's tissue, resulting in lower risks for the affiliated challenges. So some challenges with these developments. Immune rejection is one, where the new tissue is incompatible with the old tissue and is basically just attacked as a foreign invader. There's the genetic stability issue. Stem cells are just genetically unstable, kind of as a natural extension of their properties. And also tumors become a big issue. The cells are induced to replicate at a less regulated rate. This, coupled with the genetic instability, acts as a risk factor for development of tumors. This is seen in induction of pluripotent stem cells. the technology has to be handled with care in order to avoid the stem cells just replicating at an unregulated rate and basically creating a big tumor. All right, that's all. So goodbye and thanks for watching.